Assalamualaikum. This is Dr. Mohammad Noman, resident urology. SIUT. The topic of presentation is testicular germ cell tumor, meticulous aspects. And today we are going to discuss about the tumorogenic model of germ cell tumor, pathology and biology of germ cell tumor. And we will see some basic germ cell development, and from there the cellular region of germ cell tumor and extragonadal germ cell tumor, and genetics of germ cell tumor, and then the pharmacodynamics and mechanism of action of cisplatin, then apoptotic pathway, proficiency and resistance, then finally the apoptosis. The tumorogenic model of germ cell tumor, we all know that the germ cells are totipotent germ cells. They can differentiate into any cell or any tissue in our body, and dependent on three layers: endoderm, mesoderm, and ectoderm. Totipotent germ cells, if they develop normally and under normal circumstances, they develop the normal spermatocytes, which is the normal sperm inside the testis. If there is any mutation or any causative agents or other things, the totipotent germ cells can differentiate into adenoma. It is another type. It is one type of testicular germ cell tumor, or it can develop the embryonal carcinoma. Embryonal carcinoma is a broad term. It is a totipotent tumor cell. In embryonal carcinoma, we have two factors or two paths. If it is this tumor develops during the mutation or any neoplasia from the intraembryonic differentiation, it will lead to the teratoma. Intraembryonic in the embryonic the differentiation which happens in the embryo. Like teratoma, it has all the parts of three germ layers, which are destined to develop in the three particular sectors in the future. Or if it happens during the extraembryonic differentiation, like the yolk sac and the chorionic layer which are surrounding that embryo. If the yolk sac, it is in the yolk sac, it is called a yolk sac tumor, or if it involves the chorionic layers, it will be called as choriocarcinoma or trophoblastic path. Now the pathogenesis and biology. The carcinogenesis of germ cell tumor is poorly understood. As noted earlier, testicular germ cell tumor develops from a precursor lesion, intratubular germ cell neoplasia. It appears to develop from arrested primordial germ cells or gonocytes that fail to differentiate into pre-spermatogonia. These cells are thought to lay dormant until the puberty, when they are stimulated by the increased testosterone level. In the coming slide, there is a chain where I will show about the development of normal spermatocytes, spermatogonia, and then the spermatids. If there is any mutation or any arrest of growth in any lineage, and that cell is dormant until puberty inside the testes or area where the testicular tumor is going to present in future, and during the puberty, under the surge of testosterone, these mutant cells, they start to grow and they will form the tumor. The increased incidence of testis cancer that started in first half of 20th century has been accompanied by the increased incidence of other male reproductive disorders such as hypospadiasis, clitoridism, and subfertility. These findings led to the hypothesis that testis cancer and these other disorders all resulted from a testicular dysgenesis syndrome, which results from the environmental and lifestyle factors and genetic susceptibility. The specific environmental lifestyle factors have not been defined. There are four things in testicular dysgenesis syndrome. Number one, germ cell tumor. Number two, hypospadiasis. Number three, clitoridism. And number four, subfertility. There is a hypothesis that the increased prenatal estrogen exposure has been hypothesized as a risk factor, but this is controversial. There is a stronger evidence that reduction in androgen activity can result in features of testicular dysgenesis syndrome, including clitoridism, hypospadiasis, and impaired spermatogenesis. But a direct link between reduced androgen signaling and intratubular germ cell neoplasia remains hypothetical. The evidence for genetic factors include that the clustering of testis cancer in some families, the extreme difference in the rate of testis cancer in black and white Americans, and the findings of susceptibility loci on chromosome 5, 6, and 12 in case control studies. These are studies which are being held in developed countries. In addition, specific polymorphism of certain genes, including the gene encoding KIT ligand, have been associated with an increased risk of testis cancer. This KIT ligand, KIT pathway, it is an important and necessary pathway for the development of the germ cells. Monocytes depend on KIT ligand for survival, and the gene for this protein is located on short arm of chromosome 12. An increased number of copies of genetic material from short arm of chromosome 12 is a universal finding in testicular and extragonadal genital tumor. When there is a mutation or neoplasia or tumor, the tumor cells have to survive. When there is a mutation in CKIT ligand or genes producing this ligand and this survival thing for the germ cell tumor, there will be the increase in the amount of the copies of this ligand. And this is a product of a gene which depends on short arm of chromosome 12. And in both testicular and extragonadal germ cell tumor, we will get a lot of copies of this KIT ligand. Of germ cell tumor, 70 to 80% have an extra copy of chromosome 12 in the form of an isochromosome 12p, whereas the remainder shows gain of 12p sequence detectable with fluorescence in situ hybridization. There are two things. There may be an isochromosome of chromosome 12, or there may be the increase in sequencing in chromosome 12p for the production of its product KIT ligand. There is a connection between mutation or polymorphism in CKIT ligand and germ cell tumor. There has a biological plausibility. One of the most striking features of germ cell tumor is their sensitivity to cisplatin-based chemotherapy, which enables cure in most patients with widely metastatic disease. In the era before cisplatin-based chemotherapy, the cure rate of patients with advanced germ cell tumor was 5 to 10%. And at present, the long-term survival for men with metastatic germ cell tumor is 80 to 90%. This is a 5-year survival rate. The specific biologic basis of this acute vulnerability to chemotherapy is incompletely understood, but is thought to derive from the close relationship between germ cell tumors and embryonal stem cells and gonophytes, which have a low threshold for undergoing apoptosis in response to DNA damage. This is a basic point why the germ cell tumors are prone to damage after cisplatin-based chemotherapy induction, because they have low threshold for undergoing apoptosis in response to DNA damage. Gene expression analysis has found an upregulation of.
Noman, can you explain this thing, the last point, the last sentence, once more?
In the human,
for instance, what I understand is, if a cell is in a multiplicative process, the more rapid the multiplication process, the higher the vulnerability of that cell to be damaged by any agent. For instance, damage to DNA always occur when the DNA is actually in the form of going through different stages in the cycle of mitosis, for instance. So this is the kind of knowledge that we have from earlier days in physiology, when we have studied physiology. So what do you say by this thing? Undergoing low threshold for undergoing apoptosis. What do you mean by it?
So when the cell, like the germ cells, which are totipotent, which are rapidly dividing cells, then as you said, the multiplicative phase, when they continue to divide, and there is the one mitosis and two meiosis, and during this phase the cell has to create or make its own genetic material as fast, because it is dividing as a rapid speed, because embryonal stem cells and gonocytes both are totipotent cells. They are dividing in rapid phase. And germ cell tumors, this tumor is basically the mutation in this series in the production of spermatocytes. And when there is a mutation in the genetic cells, and it is continuously dividing, the cell's own defense mechanism, which is called nucleotide excision and repair mechanism inside the DNA, that mechanism detects the change or mutation or an abnormal sequence in the DNA, in the genetic form, the nucleotide sequence, and then they detect that particular defect, and they excise that nucleotide, these nucleotides, and then there is an enzyme which repair these defects. And in gonocytes and germ cell tumors, in gonocytes, but in normal cells, this mechanism is present to remove any damaged spermatocyte or damaged cell, so that it cannot enter into the normal hierarchy. In germ cell tumors, where there is a mutation, and these cells are rapidly dividing cells, because in tumor they have overtaken the cellular or host defense mechanism. But after giving the cisplatin chemotherapy, this cisplatin is a platinum-based chemotherapeutic and alkalizing agent. It enters inside the germ cells, tumor stem cells, and it binds with these defects. And these defects, then it forms the DNA attacks. Means that this chemotherapeutic agent adheres itself to the particular part of defective DNA, and this defective DNA is no longer being
repaired
by the nucleotide excision and repair mechanism.
Right.
When this is not repaired, it then further stimulates the basic mechanism of apoptosis. So this is called that these cells have no threshold for undergoing apoptosis, which is a programmed cell death.
Right.
In normal cells, the cell die. In tumor cells, after giving this agent, it has a very low threshold, because the nucleotide excision and repair mechanism cannot repair the DNA attacks formed by this chemotherapeutic.
Right. So I want to address to all my friends who are listening to us. This is a very important concept. You see, cisplatin has got a platinum sort of a thing, and again another compound which is a chemotherapeutic compound. What this platinum is doing is something what is explained by Noman rightly, that it sort of inhibits or slightly what we call, it doesn't allow the repair process, which is a normal process in a normal cell, to happen. So it actually sort of hinders or gives an obstacle to this repair process, and that is the reason why the chemotherapeutic drug is able to act more precisely and more accurately and more efficiently when platinum is there around. Is that so, Naman?
Yes, sir.
So are you listening to us, all other people?
Yes, sir, we are listening.
So,
yes, sir,
right. So is it clear, or do you want to say to us that we have to add something to your knowledge? It is clear?
Basic mechanisms are समझ आ गया.
Right. Okay. So please proceed, Noman.
Thanks. Now we are going to talk about some genes. Genetic expression or gene expression analysis has found an upregulation of numerous genes that facilitate apoptosis. Now I'm going to point out the genes which facilitates the success of cisplatin-based chemotherapy in the testicular germ cell tumor. Gene expression analysis found upregulation of numerous genes that facilitate apoptosis, including FAS ligand, IL, and PAX, whereas BCL2 is downregulated. Now, in a cell, there is a group of protein called BCL family. And in BCL family, there are two things. The BCL families, there are receptor proteins and effector proteins and inhibiting proteins. The inhibiting protein which inhibits the process of apoptosis is the BCL2 protein, and the protein which promotes the apoptosis is the PAX and PAK protein and FASL ligand. Expression pattern of genes controlling the G1S phase break point in germ cell tumor appears to promote the induction of apoptosis. G1S phase, in the cell cycle there are phases: G1 phase, S phase, synthetic phase, and then G2 phase and mitosis. In addition, germ cell tumor lack transporters to export cisplatin from cell and have a reduced ability to repair cisplatin-induced DNA damage. In the previous lecture, when we have been told that the germ cell tumor, when the cisplatin enters inside a cell, its effectivity will remain unless it is inside that particular cell. Once it is get pushed by the cell outside the cell boundaries, it will have no effect on the cell. And in germ cell tumors, they don't have the pusher, or they don't have any exporter to export the cisplatin out of the system. And they have reduced ability to repair cisplatin-induced DNA damage, which I will explain in a later slide why it has the reduced ability. Germ cell tumor have high intrinsic level of wild type p53 protein, which plays a role in mediating cell cycle arrest and apoptosis. And p53 mutation in germ cell tumors are rare, yet differences have not been consistently found in p53 status when comparing chemo-sensitive and chemo-resistant. Now, p53 protein, it is the well-studied and very highly studied p53 protein inside the human genome, because it takes part, or it has found its role in numerous processes and numerous carcinomatous processes. Germ cell tumors, they have the high level of wild type of p53. There are two types, and wild type and mutant type. Germ cell tumors, they have wild type for p53, and this protein promotes them to undergo the apoptosis. And some minute cells, they have the mutant type of p53. There is a mutation in TP53 gene, and so that these cells have the mutation in the protein which causes them to undergo the apoptosis or cell cycle arrest. So that these particular cells, they might have the chemo resistance, which I will explain in the further slides, which is an hypothesis. Similarly, expression of anti-apoptotic protein BCL2 is low in germ cell tumor, but BCL2 levels do not distinguish chemosensitive and chemoresistant cell lines. A small fraction of germ cell tumors are resistant to chemotherapy, but the basis of the resistance remains obscure. Impaired DNA mismatch repair and activating BRAF mutations have been associated with treatment failure. BRAF pathway, it is also a very necessary and important pathway for the survival and growth of the germ cells. Now, after this complex discussion, there is I would like to just go through the normal germ cell development. Germ cells arising from the embryonic ectoderm, the primordial germ cells, is first recognized during gastrulation based on its expression of alkaline phosphatase. The primary germ cells subsequently migrate to the genital ridge, where they further develop into the gender distinct gonads, a process dependent upon the presence of or absence of stromal expression of SRY, sex-determining region on Y chromosome. In males, primordial germ cells in combination with sertolized cells form seminiferous cords, which along with Leydig cells subsequently organize into the embryonic gonads. by about 2 months of gestation age. This is the flow sheet having very in capital letters the genetic pathways or which are important for the development. The primordial germ cells undergo the mitosis. In mitosis, the number of chromosomes they doesn't change. Monocytes they develop into the two types, dark type and pale type, and they have different functions. Dark type and then this is the leptotene, zygote, pachytene. These are the stages of the cell cycle. For example, zygote and pachytene is second and third stage of prophase, and then the diploteine spermatocytes. And finally, after the two stages of meiosis, they form the round type of spermatids, and then the spermiogenesis, elongated spermatids is finally formed. This is a normal path. And there's letters which are written in as we have heard these genes in just KIT ligand pathway, mega pathway. This is a normal flow sheet. Spermatogenesis. In testes, seminiferous tubules, these cells are present in a sequence. The primordial germ cells, which are inside the embryo in the fetal life. Then they try to go to spermatogenesis, and mitotic region they have the 4n number of chromosomes, and the primary spermatocytes also has 4n number of chromosomes. And first meiotic division, the primary spermatocyte they form the two secondary spermatocytes, and this is a meiosis division, so the number of chromosomes will get half, 4n into 2 and 2n each. And after the second division, we have the spermatocyte which is n and half number of the normal cell. And
so
after the further modification.
Noman, what is the normal time taken by the primordial germ cell to go different stages up to the stage of spermatid? What is the normal time cycle?
Sir,
yes, Shersha.
Sir,
Shersha wants to say something.
So it's 70 days.
Right. So it is basically between 65 and 70. Some say on an average 67 days. So you are very right. Yes, please, Noman.
Thank you. Now, cell of origin of germ cell tumor. There are two models. One is proposed by Sakabeck and colleagues, postulates that primordial germ cells or monocytes, while still in utero, but after reaching to the genital ridge, initiates abnormal cell proliferation under the direction of KIT pathway activation, leading to the intratubular germ cell neoplasia in situ, unknown region. This pre-malignant lesion remains dormant until puberty, when under stimulation from gonadotrophins, undergoes further transformation and acquires extra copies of 12p chromosome and evolves into the invasive germ cell tumor. This is one hypothesis. And another model proposed by Shankati and Halford suggests that the zygote/pachytene spermatocyte with a 4n DNA content is the cell of origin. The error-prone homologous recombination at this stage of germ cell development acquire acquisition of increased 12p copy number, which leads to the aberrant gene expression, increased mitosis, re-establishment of pluripotency, and genomic instability that supports the malignant transformation of germ cell tumor.
So, Noman, Noman, please, you want to say that the potential to become an invasive germ cell tumor later in life, actually, the information is there in at just in utero.
Yes, sir, in utero. According to this model, the information is in utero.
Right. Okay. Please.
And now there are the hypotheses or models for the origin of extragonadal germ cell tumor. The two mechanisms have been proposed to explain how germ cell tumors of extragonadal primary sites other than the retroperitoneum arise. The conventional hypothesis suggests that the primordial germ cells or gonocytes get left behind while migrating through the embryo to the genital ridge and eventually transform. While this model is easy to conceptualize, misplaced germ cells at the primordial germ cells or gonocyte states have never been identified in developing human embryo. Finally, extragonadal germ cell tumors have been identified to have chromosomal changes highly similar to the gonadal germ cell tumors with increased copy of chromosomes 12p. These alterations are thought to be acquired later in germ cell development, meiosis of primary spermatocytes, then during the migratory stage of gonocytes, raising the doubt to this theory and instead supporting a common cell origin of gonadal and extragonadal germ cell tumor. This is a one theory, one explanation of development of extragonadal germ cell tumor. And an alternative explanation involves the potential of transformed germ cells to undergo reverse migration to the mediastinum or pineal gland, where stromal environments left over from embryological development could remain fertile for transformed germ cell proliferation. One is saying that the some primordial germ cells are left behind in these areas, in the mediastinum or in that part of the embryo, and they remain there and are not being able to identify due to the human embryo they have, because they have the normal the same features like the increased number of 12p chromosome. So we can say that this is in one theory, and in another theory it says that they migrate, reverse migration from the normal testicles, they descend to the scrotum, but in reverse migration they ascend toward the mediastinum or pineal gland. And the concept of retroperitoneal primary tumor remains controversial, as many believe these cases to represent metastatic lesions from testicular tumors that were not able to be identified by ultrasound or objective. Changes in the testicular parenchyma, where a tumor has undergone out species, and could explain the failure to identify a gonadal primary tumor in some cases of solitary retroperitoneal masses. And this concept is difficult in this slide, that the metastatic lesion, for example, patient is having a testicular intratubular germ cell neoplasia inside the seminiferous tubules, and the concept behind the retroperitoneal primary tumor, because the primary focus is inside the testis and it has a metastatic or point inside the retroperitoneum. This is which we have primarily diagnosed as retroperitoneal primary tumor, but the primary tumor is inside the testis, and we are not able to identify that with found in the tubular germ cell neoplasia. It is inside the cell, it has not invaded the anti-basal membrane.
So you want to say that anything in the retroperitoneum is actually a metastatic focus and the primary focus is burned out?
So exactly, it can be secondary focus, or it might be the primary, but we were not able, according to this hypothesis, we cannot say directly that it is actually inside the until unless we have done the particular investigation to find out that there is no primary focus in the both testes.
Suppose what happens is during migration of testicular tissue from the genital ridge to the scrotum, they are left in that path somewhere in life. That might happen.
Yes, sir. So that might happen. Number one, they are left behind the germ cell tumor. They are left behind. Number two, there is the migration, reverse migration. It can, if it happens to the retroperitoneum, then that might undergo the retroperitoneal primary germ cell tumor. And number three, according to this slide, number three, there is a silent focus in the testes, which is about not or intratubular germ cell neoplasia. We cannot differentiate unless we get the box of that particular region. And the retroperitoneal tumor is actually the metastasis of that silent testicular tumor. Then, according to this slide, there are three concepts behind this retroperitoneal tumor.
Okay.
Now, genetics of germ cell tumor. Like we have studied the genetics previous slides, but now we will summarize the things the genetics in detail view. In the germ cell tumors, we have first we have studied in previous slide that it is a universal finding to have increased number of copies of chromosome 12p or increased genetic sequencing in chromosome 12 in all testicular germ cells or extragonadal germ cells. So, number one, the gain of chromosome 12p. Number two, chromosomal changes which are other than the chromosome 12. They are also identified after the integration of intratubular germ cell nuclei. There is mutation in the chromosome 1, 5, 7, 8, 12p and 10, and the loss of DNA content from chromosome 18. This is academic, just for the over knowledge. Adjacent invasive tumors also exhibit many of these changes, but in addition, frequently had the gains of portions of chromosome 2, 3, 6, 13, Q, 14, Q, 17, and 20, 21, and losses of portions of chromosome 1, 4, 6, 9, 11, 13, 19. And in some cases, there is the gain, and some cases there is a loss. There is not only a single chromosome 12 or 16. There are this is the tumor with of any type of any origin are mostly under when the mutation in the different set of genes in different set of chromosomes, not a particular single gene. There are two basic things: the gain of chromosome 12p and other chromosomes, and the mutations. In contrast to most malignancies, germ cell tumors are believed to contain relatively few driver mutations. However, mutations in KRAS pathway, KIT lichen or KIT pathway, or SMAD4 have been identified in some germ cell tumors. They have been proposed as important in germ cell transformation. KIT is perhaps the most well-studied of these genes. The number 4 factor is genetics and pathobiology of chemosensitivity and resistance. The introduction of cisplatin in the late 1970s radically changed the outlook of post-pubertal men with advanced germ cell tumors, increasing the complete remission rate from 25 to nearly 80%. And the transformation from a platinum sensitive to platinum resistance phenotype likely depend on the change in several intracellular pathways, including those involved in cellular response to DNA damage and those involved in the apoptosis, differentiation, and cell growth. And several studies have demonstrated difference in immunohistochemical staining of markers of cell proliferation and apoptosis between different germ cell tumor histologies. For example, in one study, the embryonal cell carcinoma were demonstrated to have the highest rate of apoptosis and negative staining for BCL2. And the BCL2 is an anti-apoptotic protein. And in contrast, a mature teratoma, which are highly chemo-resistant, has very low level of apoptosis, and they have they positively stained for BCL2 protein. The cells which are highly chemosensitive, like embryonal carcinoma, they have increased amount of apoptosis and decreased amount of or no amount staining for anti-apoptotic BCL2 gene. And the teratoma, we all know teratoma, it is chemo-resistant. It has very low level of apoptosis, and it has positive staining for anti-apoptotic. This is another genetic factor for its chemo-resistance and chemosensitivity. Now, the cisplatin, pharmacodynamics. Cisplatin is anti-neoplastic in the class of alkylating agents and is used to treat various forms of cancer. Alkylating agents are so named because of their ability to add alkyl group to many of electronegative groups under conditions present in cells. They stop tumor growth by cross-linking guanine bases in DNA double helix strands directly attacking the DNA, and this makes the strands unable to uncoil and separate. Because this uncoiling and separation is necessary for replication. When there is no uncoiling and separation, the cell can no longer divide, so there is a arrest of cellular growth. In addition, these drugs add methyl or other alkyl group onto the molecule where they do not belong, which in turn inhibits their correct utilization by base pairing, cause of miscoating of DNA. And alkylating agents are cell cycle, they are non-suppressive. They don't have they don't particularly act on a single checkpoint inhibitor as a single checkpoint. They have no specificity for the cell cycle, the phases of cell cycle. Alkylating agent works by three different mechanisms, all of which achieve the same end result: disruption of DNA function and cell death. What is the mechanism of action of cisplatin? It is believed to kill cancer cells by binding to DNA and interfering its repair mechanism, eventually leading to the cell death. The first step in process after the cisplatin molecule penetrates the cell membrane intact is for a molecule of water to replace one of the chloride ions. And just from the chemistry side, the cisplatin, which has the platinum inside it and platinum is transitional element, it changes its valency according to the environment. But sometimes it's 2 and sometimes it is 4. And in this alkylating agent as a chemotherapeutic drug, we have cisplatin platinum with 4 valency. On two sides, there is I will show you the structure later.
So is it plus 4? Is it plus 4 or minus 4?
Sir, it is plus.
Right.
Cisplatin is believed to. I will make sure this is plus 4 minus 4 later.
It is plus 4. It is plus 4. I was testing you.
Cisplatin is believed to kill cancer by binding to DNA, interfering the DNA, and as I previously told that there is a nucleotide exchange and repair mechanism. So nucleotide exchange and repair mechanism does not able to repair the defect or adducts produced by this chemotherapeutic agent in the germ cells. The resulting structure can then bind to a single nitrogen on a DNA nucleotide. The second chloride is replaced by an first I will show you the structure. This is a cisplatinum structure, platinum, and on the two sides there is a chloride group attached, and on the other two sides there is the NH3 group is attached. And there are two mechanisms. One is there might be a receptor for the cisplatin receptor, for example, or there might be a passive diffusion, because inside the cell there will be no cisplatin before the induction, so it will be passively diffused inside the germ cell. Good affinity for the tumor cell. So inside the cells, they undergo the hydrolysis, change of one chloride for the hydroxyl group, another chloride for the hydroxyl group. Then this pair is entered into the nucleus and get attached to the mutant or tumorous part of the DNA and form the DNA attack. So when they form the DNA attack, there will be no repair. And if there is no repair, then the DNA, due to its own ability to promote or propagate the pro-apoptotic genes and apoptotic genes and inhibiting the anti-apoptotic genes, they undergo the process of apoptosis, and hence there is a cell growth arrest and then further death of the particular cells. So now the theory: the second chloride is replaced by another water molecule, and the platinum binds to a second nucleotide. Binding studies of cisplatin with DNA have indicated the preference of nitrogen 7 on the two adjacent guanine bases of the same strand. It also binds to adenine and across strands to a lesser extent. The cisplatin DNA complex attracts the attention of HMG high molecular group and other DNA repair mechanisms, which become irreversibly bound. The resulting distortion to the shape of DNA prevents the effective repair. So this is a table. It is a proposed mechanism to explain the extreme sensitivity of germ cell tumor to cisplatin and why there is the development of resistance in some of the germ cell tumor. Number one process is the DNA repair process. I have already explained the DNA repair process, because the DNA cannot repair the damage caused by the cisplatin. Number two, the apoptosis, and there the high plausibility or high proficiency of apoptosis in germ cells, and upregulation of apoptotic gene and downregulation of anti-apoptotic gene. And number then TP53. TP53 is the gene which produces the wild type of P53 protein in germ cell tumor. Firstly, this gene is frequently mutated in other carcinoma. And germ cell tumor resistance to cisplatin is associated with the mutation in TP53 and P21. Germ cell tumor, they usually have low level of cytoplasmic P21 and high level are associated with cisplatin resistance. Is a isochromosome 21. It is a cyclin-dependent kinase responsible for inducing cell cycle arrest at the G1S checkpoint to allow for the DNA repair. So this P21 helps in the DNA repair. So when there will be the high level of DNA repair, so there is no tumorous activity, and when this protein is very low, this might result in the DNA damage or neoplastic ability. Then the differentiation, the most common germ cell tumor histologies, they exist in undifferentiated state, such as seminoma and abdominal carcinoma. Differentiation such as teratoma leads to the platinum resistance. When there is a proper differentiation, like the teratoma, all three germ cells, they have differentiated into their respective structures. Once they are differentiated, they have achieved their final stage. There is no room for cisplatin or platinum-based agent to act upon it for the resistance, because they have they get removed themselves from their totipotency. Driver mutation, there are some mutations, BRAF mutation proposed but not validated, which is cisplatin resistance, P53 mutation, which have a role we have studied, and few other mutations in germ cell tumor like KRAS, HRAS, and AKT. Now, apoptotic pathway, proficiency, and resistance. In addition to mutation in TP53, aberration in other part of apoptotic signaling could also lead to the resistance. For example, a cell line with inability to activate caspase 9 maintained cisplatin resistance, independent of whether TP53 was wild or not. Number one. Number two, another study found that upon exposure to cisplatin, the sensitive germ cell tumor cell line displayed an increased expression of FAS ligand and recruitment of FADD death receptors and caspase 8 to FAS, whereas the resistant line did not get the exposure. These Austrian colleagues found sensitive germ cell tumor lines to have decreased cytoplasmic P21 as compared to the resistant cell line. It is well explained in the previous table. The silencing, how to increase the sensitivity of resistance to tumor. Silencing of P21 or manipulation of increased P21 shuttling from cytoplasmic to the nucleus increase the apoptosis and they restore the cisplatin sensitivity of the resistant germ cell tumor. For example, the P21, when it is present in high quantity in the cytoplasm, so it can identify the DNA defect, and it stimulates the DNA repair mechanism and it repairs that defect, so there is no tumor. So in resistant cell, there is high quantity of P21. So the germ cell tumors, when in germ cell tumor cells, when there is a high quantity of P21, they have the resistance to the cisplatin. By silencing this P21 or changing its position from cytoplasmic for transporting it from cytoplasm to the nucleus, it helps to fight with the resistance of these resistant germ cell tumor and make them sensitive to the cisplatin. This is another hypothesis. This is a flow sheet of the chemotherapy of the germ cell. First of all, we have to get the cisplatin inside the cell and it forms the DNA attacks. And DNA attacks, after the damage recognition, they underwent the apoptosis. And these points which have shown the T, like uptake, effect, defect, increased drug inactivation, and these are other the inhibiting agents. If these factors are present, then there will be no apoptosis, then there will be the resistance. Like loss of p53, defect in MMR membrane repair. Number two, overexpression of certain genes, and increase in if there is increase in DNA repair, there will be the resistance to the chemotherapy. If the caspases are not activated, there will be the resistance. They will inhibit this apoptosis mechanism, and the neoplastic event will continue to grow. And if there is increased BCL2 anti-apoptotic gene, it will inhibit the apoptosis. Down regulation of pro-apoptotic bags or bad protein, they will inhibit. So this is the combined picture to show all the inhibiting factors, and if these inhibiting factors are not present, they are the promoters. So now toward the last slide, what is apoptosis and what are the mechanism of apoptosis? Apoptosis is a programmed cell death. So there are two pathways for apoptosis. One is intrinsic pathway and one is extrinsic pathway. Intrinsic pathway, it involves the mitochondrium, and extrinsic pathway, it is a death receptor pathway, FAS ligand and FA double. When there is any injury, cell occur to cell, like when there is a survival signal, inavailability, growth factor withdrawal, DNA damage by the radiation, by the toxins, by the free radicals, by the chemotherapeutic agents, or if there is a protein misfolding, they will activate the BCL2 family sensors. BCL2 family sensors are the pro-apoptotic genes. They are BAX and BAC protein. They enter into the mitochondria and they changes its configuration. And from mitochondria, the cytochrome C and other pro-apoptotic protein, they get leaked into the cytoplasm. When they enter, when there is a cytochrome C in the cytoplasm, it activates the caspases, and there is a whole chain of caspases. When the caspases are the end part of caspases are also known as the executioner caspases. These caspases proteins, they activates the certain enzymes. For example, endonucleases. Once endonuclease is activated, it will damage or do the DNA fragmentation, because it is the endonucleases inside the nucleus, and it also activates the other lytic enzymes so that it can break down the cytoskeleton of the whole cell without damaging the cell membrane. So when the whole inside the organelles of cell are being damaged or being distorted, the cell membrane forms the small flaps outside it, and it accumulates a little debris and get itself separated from the that cell. And so hence the whole cell is being changed into the small flaps and disintegrated. So, and they form the particular cytokines which attracts the phagocytes from the adjacent structures, adjacent tissue, and these phagocytes, macrophages, they engulf this particular cellular flap and make them disintegrate. So this is the end point of any pathology in the cell. Thanks.
So, Numan, if you just go to this slide, the last slide, just put up the last
This
slide.
one?
Yes, this one. As I can see here, there are two pathways. One is intrinsic, the other one is extrinsic, and it is nicely done diagrammatically. It is explaining a lot. But if it is programmed, programmed means, for instance, I've got a red blood cell which has got a life of probably 100 days and after, or 120 days, and after 120 days it will be wiped out, and then it will be taken up by my spleen, and then the spleen will just break it down into constituents, into separate constituents. Now, if that is something which is programmed, then I don't need anything which can damage, for instance, radiation, toxins, free radicals. So what happens when automatically something turns on? What in this whole diagram, programming comes in?
So, sir, there is a cell survival signal outside on the cell membrane of any cell, like, up to my understanding.
Mm-hmm.
If there is a absence of that survival signal, as it is mentioned under the slide heading of mitochondrial intrinsic pathway, cell injury, growth factor withdrawal. When on any cell there is a signal, or this is like a bulb or which is bulging around the cell, and when there is that thing, it means that the cell is alive, or the cell has to live until unless there is that particular signal. If there is no growth factor or there is no cell survival protein attached to that cell, so it will not live, or it will cause cell destruction. The mechanism of cell suicide, which is present in there, like as you said in RBC. And second point is in the extrinsic pathway, there is a ligand, FAS ligand. So it has to get attached to the FADD death receptors. There are two things on the cell membrane of cell. Number one is the survival signal or survival factor. Number two, the ligand which has to attach itself to the death receptor. So number one, when there is a deficiency of growth factor or survival signal. Number two, when the FAS ligand attach itself to the death receptor to activate the suicidal mechanism. These both mechanisms, these are the programmed mechanisms to induce the suicide or programmed cell death of particular cell. When these two mechanisms are not present, no matter how much mutation or how much proliferation of tumorogenic element inside the cell cannot do this programming, and there may be the two pathways. Number one, it might undergo the proliferation to form any invasive or malignant neoplastic process, or number two, it can damage the cell membrane, and all the enzymes or all the contents inside the organelles which are dangerous to the nucleotides and cell membrane, they will get expelled from that particular cell outside the surrounding tissue. This is called programming, because it only kill or it only get the death of that particular cell which is no longer required without damaging the surrounding structure. So this is programming up to my understanding. I might.
No, you are quite right. When we use the term programming, then we have to know where this programming is actually being done and where this programming is being stored. Now, DNA is not only present in the nucleus, but also it is present in the mitochondria. So, in your intrinsic pathway, you see there is a very central point for mitochondria to act. For instance, cytochrome C and other pro-apoptotic proteins. So there is a balance between two processes. One is pro-apoptotic processes, and the other one is the anti-apoptotic processes. Now, in today's world, when we talk about something of the nature of chemotherapy, then there is a class of drugs which are actually pro-apoptotic. What they do is they sort of trigger on those processes which are actually causing cell death. So this is a very interesting the mechanism and the scheme that has been shown here in this diagram. But there are other players also, and we have to take care of them also in this total picture. This is one way of looking at it, but for a novice or someone who has just started to think about this subject, it is fair to understand and to talk to that person, just to remember that these are the two processes going on in every cell of the body, right from birth till the whole body dies. And this balance is between anti-apoptotic and pro-apoptotic processes. So this programming is actually in the DNA, but that DNA can be there in many places, and whenever that DNA is triggered on, there are this information which is stored there, and that has got its own implications. So very nice presentation. All our colleagues, this might seem a bit too exhaustive and too detailed. But you see, we have to change gears. We have to change gears in order to understand a subject properly. And this is the actual beauty and flavor of what we call research. So an MS program is actually a research-based program. You have to increase your capacity. You have to be a dreamer, for instance. If somebody asks me, I'm not an MS, I haven't got an MS degree, but if somebody asks me what is an MS program, then for me it's a dream. So unless or until every one of you has got a dream, um, you can be or cannot be a researcher. So a researcher is the one who has got a dream, which might be conceived or which might not be conceived. That's again the beauty of this whole process. Sometimes at the end of your research period, you will say that I have started with one notion in mind that might be the hypothesis, but it hasn't been proved yet. So that's how the whole process of science goes on. It doesn't end with something which is known as preconceived idea. It might end at some point which hasn't been conceived yet, a new thing. So the forum is open for any discussion, any point that you want to share with us. Everybody can speak. Yes, Rafe, you want to say something, or anybody who wants to say something? Yes, Mohsen, you want to say something?
No, sir.
Right. Okay. So today we have been a little late to start with, but we have got, I think, 15 people joining in, and I had one request from Bangladesh also. I don't know whether he has joined us or not. He was with us as an MS candidate in the early part of this century, and he did his MS from SIUT. Now he is working as associate professor. His name is Nazmuddin Muhammad Arif, and he is an associate professor in Dhaka. So he had this knowledge of our meeting and lectures, so he wanted to join in. Probably he joins tomorrow or day after tomorrow, so he'll be with us. He is very interested in this kind of activity. So thank you, Numan, for enlightening us, and this has been a very good explanation from your side about your subject, and we'll move on. So for all those who want to just carry on this process of the second round presentation, we'll be expecting a lot detail from you in the second round. And again, as I have said earlier on, we are changing gears. We are changing gears to understand more properly each and every aspect of urology. So let's hope that we'll get something in a better way, and that can increase your experience and make your life a little interesting also when you do a second round presentation. So from Monday, I think it will be Rafe to start with.
Yes, I will, sure.
Okay. So meet you Monday. Thank you. Hafiz.